to the second day of the 3-day International Conference on Biotechnological Applications in Ayurveda and Biomedical Sciences and the annual meet of the Society for Biotechnologists India, SBTI. We hope you had a productive and wonderful day one of the conference with an insightful discussions and valuable connections at our 3-day International Conference on Biotechnological Applications in Ayurveda and Biomedical Sciences. So we are all feeling refreshed and ready for another exciting and insightful day ahead. On behalf of the organizing team, it is my pleasure to welcome you all to the second day of the International Conference. As we step into another exciting day of knowledge, sharing and collaboration, we look forward to engaging sessions, thought-provoking dialogues and meaningful interactions. We'll commence today's beginning of the day with a keynote address by Professor Dr. Ramesh Sharma sir. So the keynote address, Dr. C.S. Paulo's Oration Award keynote address, we are to begin. So a very good morning, once again, a very good morning and a warm welcome to all of you. And it is the most interesting session of the day, the keynote address. It is an absolute honor to introduce our esteemed speaker, Professor Dr. Ramesh Sharma sir, former Head and Dean, School of Life Sciences, Northeastern Hill University, NEHU, Shillong. Before we invite him to deliver his address, kindly allow me to share a brief introduction about sir. Professor Dr. Ramesh Sharma is a distinguished academician and researcher in the field of biotechnology, currently serving as a professor in the Department of Biotechnology at Northeastern Hill University, NEHU, India. With an illustrious career spanning over several decades, sir has significantly contributed to the advancement of biotechnology, particularly in the areas of molecular biology, genetic engineering, and applied biotechnology. Sir's research has been instrumental in developing novel biotechnological applications with implications for agriculture, healthcare, and environmental sustainability. Professor Sharma's contribution to scientific research is widely recognized with his work published in the high-impact international journals and numerous citations reflecting its academic significance. Sir's research endeavors have focused on genomic studies, bioprocess technology, and the application of molecular techniques in plant and microbial biotechnology. His dedication to pioneering research has positioned him as a leading expert in the field. In recognition to his outstanding contribution, Professor Sharma has received several prestigious national and international awards. His accolades include Excellence Award for Research Innovation, fellowships from renowned scientific bodies, and recognition for his contribution to biotechnology education and policy development. These honors underscore Sir's commitment to scientific advancement and his influence in shaping biotechnology research in India and beyond. Apart from Sir's research and academic achievement, Professor Sharma has been actively engaged in mentoring young scientists and scholars. He has successfully guided several doctoral and postdoctoral research, many of whom have gone on to make significant contributions in the academia and industry. Sir's expertise is sought after in various scientific commitments, editorial boards, and policy-making initiatives, where he plays a crucial role in shaping biotechnology research and education. Professor Sharma's unwavering commitment to biotechnological advancement, academic excellence, and mentorship continues to shape the future of biotechnology research. Sir's contribution to the field remains invaluable, reinforcing the critical role of biotechnology in addressing contemporary global challenges. And today, Sir is before us to deliver his Dr. C.S. Paulo's Oration Award keynote address. And the topic for the lecture today is Role of Ayurveda in Health and Longevity. May I now invite Sir for the lecture. And sir, we are now eager to hear your thoughts and wisdom.
Very good morning. Is it okay? Very good morning to each one of you present here. Before I start my presentation today, I would like to profusely thank Professor Bijian and his team of Society of Biotechnologists India for choosing me to this Oration Award lecture. I'm privileged to have this award uh, because I also knew Professor C.S. Paulos. Can I have the first slide, please? Professor C.S. Paulos, when he was doing his PhD and almost finished his research work, and I had entered. And I had seen him for almost 6 months to one year. Then he left the lab from Banaras. He had done excellent work during his PhD on neurotransmitters, brain function, and on aging. And I continued after my PhD, and I think we met after 25 years after that. And then he used to come to Shillong, and we used to have good interactions. I had visited CUST, the Cochin University of Science and Technology. So I thank profusely all the colleagues of the Department of Cochin University of Science and Technology. I also thank the organizers, particularly the Sunil and Amit, for having invited me to this presentation today. It is my privilege to speak before you. I have designed my lectures something for everyone, for students as well, because the topic which I have chosen may not concern the back side today, maybe front side today and back side tomorrow, because it is on aging and you are still young, so it will take time for you to age. And what are the biochemical changes that happen during aging? And what can we, if we know the reasons, then we can have some interventions to have healthy old age. The purpose is not to add years into your life, rather life into years, so that you can remain healthy as long as you survive. There is no fun of surviving for 100 years and 20 years in disability. So as long as you survive with good health and take care of your own business, so that we are not burdened to the society and to even government. And with the time, I thank, as I said, that I owe this lecture to Professor C.S. Paulos, who had been excellent worker in field of neurobiology and neurosciences, and thanks to the team. The topic I chose was on role of Ayurveda in health and longevity. I'll changing. Okay. So the aging is not a new topic. Getting old is not new. There had been frenetics. I mean, you can see in our Indian society, since ages, if somebody wishes Ayush someone, so say Ayushman Bhav. Ayushman Bhav, look to have a long life. And then somebody says Chiranjeeva Bhav, live for a long time. These are all enshrined in our culture, society for ages. Ayurveda, Ayur in Sanskrit meaning longevity. So you have Veda related to health and longevity, not from today, since our ancient time. And now, of course, Ayurvigyan, the science of longevity, which almost people have taken this in a scientific manner with the passage of time. This is not only to say our society, even in other society like Christian society. Some of you, have you heard the word Methuselah? Methuselah somewhere, anyone can raise the hand? I mean, if we go to the epics, the Christian epics, it lived for almost 969 years, and Adams for so many years. So this is not a quest for remain young. is not only in one society, all across society, people intend to live longer. And there has been a nice painting, German painting, called Fountain of Youth. If you Google search, you'll find Fountain of Youth. It has been there since time. Look at the painting where the young one will, the old one will enter and come out as young. So it has also influenced the mind of artists. Look at the nice depiction of how we age and what are the phenotypic changes that happens in our face, and see the wrinkling of his skin to that side when we become old. How do we define aging? For students particularly, aging, please keep in mind, aging is not a disease first. It is part of our lifespan as a function of time. All the changes that occur in an organism is with the passage of time is defined aging. Some people define aging as a deteriorative process, that as you age with the passage of time, we do have wear and tear, and it might lead to functional impairment. The word gerontology, I'm sure some of the student, I'm in front side, I'm sure people will know, but the student, gerontology is a branch of biology which deals with the aging of an organism. So this is a branch. However, the geriatrics is a medical specialty. I think I'm happy to tell AIMS has started a very famous place for elderly people, because if you are old and you are in the same queue as the adults, because your legs are not in good condition, you might be frail. So it is good to have a separate ward for medical specialty. So that's geriatrics. Senescence is another word. In plants, people use plant senescence, but it is interchangeably used with aging. So students can keep these terminologies in mind. As I said, aging is not a disease. It is part of our lifespan. It starts with, say, prenatal, the ovum, embryo, fetus, birth, and then you have postnatal, neonatal, infancy, childhood, adolescence, and adulthood. Adulthood has maturity, and of course, senescence, aging, and finally the death. The lifespan, the age at which an individual dies, the lifespan. And we do have variability in different species. Say rats and mice, they live for 2 to 3 years. Say if you go to human, we live for 100 years. So we do have variability among individuals. But maximum lifespan is a famous picture which I took it from net, that Madame Jean Calment, she has a record of 122 years. And she passed away in 1997, the French lady. So one can go to 122. We have many super centenarians, so-called, who have lived for 110 and beyond, but this was a recorded scenario. Of course, life expectancy, the essential lifespan, nature chose that the age on reaching reproductive maturity. Aging, as I said, sits at the crossroad. It affects almost every organ of our body, be it the brain, the renal system, the cardiovascular system. You can see here how atherosclerosis takes place, narrowing of arteries. Skeletal system, we become frail. We are not as smart as we had been, say, 30, 40 years ago. And then skin, you have wrinkling of skin wrinkles. That is how some of the pharma companies have come up with anti-aging creams. So I'm not here to vouch. There are many quacks in that, but still there are some who might have some scientific basis. Say, for example, collagen is a triple helix protein which is there in your skin, and because of those triple helix nature, because of reactive oxygen species, the amount goes up during aging. So you might have cross-linking of those collagen proteins, giving rise to wrinkling of skin. So most of those anti-aging creams, they have antioxidants in it. Let's say vitamin E. Most of those creams which you have in the market for skin, they have vitamin E, vitamin C, which can scavenge the free radicals and help in less cross-linking of protein. So that can be there. Of course, your neuroendocrine system gets altered as a function of age. And most of the physiological parameters that we have, they decline as a function of age, except the blood glucose level. You'll be surprised that if somebody doesn't pick up the diabetes, diabetes is a lifestyle disease. Whereas if you might have seen many elderly, 80 plus, their blood glucose level is exactly as it was in the young age. That is so stringently controlled. However, most of the parameters, they decline as a function of age, and most significant one is the breathing capacity. Imagine that is how we are here. If you maintain your breathing capacity, I tell you, it will do wonder to your lifespan. And that is how the yoga and yogic exercises which people are planning to do, which will help having healthy aging. So the aging, if we summarize, it's a multifactorial process. There is no single cue which you can pinpoint. So it's a multifactorial process, and it is parallel and often interacting processes. Because you do have network in the system, the connectomes, which will allow the interacting processes to govern the lifespan. And of course, it is controlled by genetics and environmental factor. Both are important. And to be frank with you, if you ask me the contribution of genetics and environmental factors, I will give, if I'm not biased, I'll give 1/3 to your genetics and 2/3 to your environment. This is very important, because a lot of time you might have seen people say that children of long-lived parents live longer. It may not be true if children don't follow the father's footsteps. So if father had worked hard and now if you start abusing your lifestyle right in the beginning, then you may not match. And contrary also, if your father had not lived long, you can still live long, because you improve upon your lifestyle, what you eat, how do you exercise, how you take care of your business. So it's always good to maintain your environment well. And of course, so the aging is dependent on interplay of both inherent, what is you are born with, and the acquired, the environmental factor, how you live with. So the hallmarks which have been now being looked into the phenomenon of aging, something which has age-associated change, like what you call markers. So what are the markers that we have? Any marker which can be included in the category of hallmark is the one which if you activate them, the aging should accelerate. And if you try to intervene them, it should decelerate. So this criteria you have to have. And today, with all these 50 years of research on aging, many theories have been proposed. Some of them have been virtually wiped off. Some are still there in the literature. But today, as a good biochemistry, molecular biology student, these are the 12 hallmarks which have been put, I think as 2023, where you have this genome instability, the DNA damage and repair. You have this telomere shortening. Telomere is the end of chromosome which gets shortened. And when it is shortened to the extent that beyond the critical level, you have the cell arrest, and the cell growth stops. So we do have epigenetic changes. We do have all these nutrient sensing pathways, mitochondrial, all these, including the dysbiosis, which has been added. Dysbiosis is the microbiome population, or the milieu of the microbiome in your intestine. So that has been added. Particularly a Japanese study, I think 4 or 5 years ago, they have centenarians and they have super centenarians. Centenarians, those who survive for 100 years, super centenarians, those who survive beyond 110. And if you take the gut microbiome of those who have lived for more than 100 years, their gut microbiome is very different than the others, those who have not lived to that period. So the dysbiosis is also added too. So we have these hallmarks. Now I'll quickly pass by to this word today, I said gerontology, I said geriatrics, I said senescence. You put all together and there is a word I'm sure some of you must have read at least, called geroscience. G-E-R-O, geroscience is a science dealing with the aging. So that's a new term which has been added to now. And what are the challenges and perspectives? We can think of health span versus lifespan. This is what I said in the beginning. We should care about how healthy we are during the later part of our lifespan rather than how many years we are surviving. Now, the next one, extending health span. The purpose has to be to extend the health span part of your lifespan rather than just the lifespan. Improve on health. So how do you do that? So if you have to improve the health parameters during aging, you have to have they improve the health behavior, particularly the food that you eat and exercise. At least these two things don't remain sedentary. So you have to have movement of your body, do exercise, whatever you do. If you, I mean, I'm not intending to suggest you to go to gym every day, but at least you work at home, you walk around, you help your parents, be active. And food that you take has to have a proper amount of food. And so living better, not just longer. This is the challenge which we have. And die young as late as possible. So that is what the priority is. So the neuroscience, and yesterday we had a lecture on personalized neuroscience, because we all as an individual, we have a different physiological and molecular setups. So one formula will not work for all of us together. So you have to devise your own system to live with it. Like even for food, somebody says, I eat 2 chapatis and I'm fine. But if somebody takes 2 chapati, may not be fine. So you take 1 or 3, whatever is suited to you. So that depends on personalized neuroscience. So now we have, so what I did for many years, almost 2 decades, I had worked on one of the interventions to have healthy aging was on dietary restriction. Eat less, live long. This is very old saying, nothing new. Eat less, live long. How much less? You should not be malnutrited, because a lot of time you must have seen the young kids, they do dieting and faint on the road. So don't do the dieting so that you can faint on road. Have the restriction to a limit of 25 to 30%, and that will be fine enough. So the dietary restriction, and the linghanam param ausadhim. This is in our Vedas, that linghanam param ausadhi. If you have restriction, it always helps. And there are many types of restrictions. Food is one. Food is one. And then of course, I took the food as a restriction. And what we did for almost 2 decades, each one of these is by my students, and thanks to them, the dietary restriction had led to reduced ROS during aging, protein carbonylation. Protein carbonylation is a oxidative modification of proteins. So when you have enough oxidative milieu, the proteins get oxidized, and particularly the lysine and arginine, they have that the side chain amino group, which amino NH2 gets converted to CO, the carbonyl group, called carbonylation. And as a function of age, you do have increased carbonylation in proteins. And if you subject the mouse model, we have worked on mouse model, so you have reduced carbonyl content, thereby meaning that relief from oxidative stress. Of course, acetylcholinesterase, which we have found is reduced by pursuing dietary restriction. The only intervention for Alzheimer's disease is the inhibitor of acetylcholinesterase. And imagine, without using pharmaceutical intervention, if you have dietary restriction, control your diet, you can have better relief from that. And of course, IGF-1, AKT, and FOXO gets reduced. NF-kappa B, as a function of age, we do have chronic inflammation goes up. So if somehow you can reduce your inflammation in the body, it will help you to have healthy aging. And we have seen dietary restriction reducing NF-kappa B. And of course, these are the ones which are enhanced to have better healthy aging. Now, coming to the topic which this part, going to the Ayurveda and the healthy aging, I'll try to have my time. So both men and women, I think some of the backside student will know, and it's good for you, that every society, women live longer than men. You can search. So there is no dispute on that. At least 3 to 5 years. Every society. developing or developed society, women live longer. Why do they live longer? Because of the physiological setups, the hormonal setups, and so on and so forth. Maybe nature chose them, the mothers, to live longer. Now, now if you look at these, the dark one here, these are the years with the health issues. That after a certain age, you do have a lot of health issues. So as I was telling you, there is no fun of living long with disability. So a lot of finding new drugs which can delay many diseases of old age. And in order to do that, we chose the nutraceuticals, which we all use in our food, whether it is turmeric powder or it is chilies or paparin or gingerol or whatever the compounds which we use, we do have nutraceuticals role in the aging process. And you can see here the very brief one I quickly pass by. If you have more ROS, then you have redox balance change, and that can have a lot of signaling molecules. Today we know redox active proteins we have, and those get altered, and of course leading to inflammation and leading to tissue damage and so on. And this is a vicious cycle. So if you have more tissue damage and chronic inflammation, you produce more ROS. So it will keep on going and having aging and age-related diseases. This is the one which I was telling you, that we do have these dietary components and phytochemical, the seventh wonder, you know, these 6 boxes, 1, 2, 3, 4, 5, 6, and the phytochemicals, the seventh one is very important, which we have been using in our society, but it needs to be done carefully now. Now, these are the components which we have gone through, we have seen. So I chose curcumin, you know, curcumin, which is active part of the turmeric powder, which we use. And you won't believe, you go to, I mean, I had been abroad for say almost 4 decades, and tremendous change. Even outside India, there are many countries which have started using. Japanese never used turmeric earlier. Now they use in their curries and many places. So as the Europe and US. So this has become very popular. And curcumin is known to all of us. It is used in inflammatory diseases, cardiovascular, and so on and so forth. Curcumin has been studied. I mean, one of the famous Indian, I mean, I feel proud to mention his name, Dr. Agrawal, Bharat Bhushan Agrawal. He is called Turmeric Man. He is in US, was in MD Anderson Medical School, had published tremendous papers in JBC and other good journals. Anyway, so what we have a lot of once you use this, it boosts the gene expression for most of these, reduce the gene expression for these oxidative parameters, and including transcription factors. Some of the enzymes are increased by glutathione S transferase, and these kinases are downregulated to give the impact of curcumin. So what I chose at that time, one of my students, I gave, you know, stress. Somebody has stress. Stress produces glucocorticoid. Glucocorticoid controls you from the stress the nature has given us. However, glucocorticoid has a lot of side effects. Although it is a life-saving drug, in fact, it was used even in corona infection because as a very good anti-inflammatory agent. Excellent. However, it has many side effects. So can we do something to potentiate the effect of glucocorticoid receptors or glucocorticoid action mechanism so that we can use less steroid with these nutraceutical components? And it turned out to be that, so this we have already seen. I'll go to, oh, sorry. So glucocorticoids, I mean, most of the students will know, glucocorticoids will enter into the cell, bind to these receptors. Receptors are in inactive conformation because it is associated with all those auxiliary proteins. So they get detached. An active receptor with hormone enters into the nucleus, bind to DNA, modulate the gene expression. However, we also know that the glucocorticoid has a non-genomic action. The one which I had told you is a genomic one, which is well established for a long time. But now we know it has a non-genomic action. Besides doing this, it also interacts with some of these players, including NF-kappa B. So glucocorticoid binds to receptor, hormone receptor complex interacts with NF-kappa B, doesn't allow NF-kappa B to go to the nucleus and give you the response. So one of the anti-inflammatory role of glucocorticoid is by suppressing the action of NF-kappa B. So I chose this, and that you can see here the normal cascade, any inflammation, NF-kappa B, and it is activated and binds to and activate a lot of pro-inflammatory genes. But the glucocorticoids, if you put glucocorticoids here, hormone receptor complex, it doesn't allow to the NF-kappa B to bind to the DNA and activate the pro-inflammatory genes. So it's very good to have an anti-inflammatory role. As I said, curcumin, I explained you what I did. The curcumin, somehow if you know how do you measure the receptors? So earlier we used to take radioactive hormone, incubate with the cell SAB, the cytosol, where you have isolated the receptor on nucleus. So the radioactive hormone will bind to the receptor, and you measure through a liquid scintillation counter, measure the receptor. What we have done, if you use curcumin, the at least 100 micromolar had enhanced the binding of receptor hormone to the receptor. So it potentiates. And same thing what we did, then at that time the bioinformatics tool came, and we tried to see, is it so? We went, in fact, in a better way at that time. Nowadays, what we do bioinformatic analysis and go to the lab work. That time we did lab work and went to bioinformatics and see whether glucocorticoid receptor interacts with curcumin or not, or curcumin interacts with the receptor or not. And it does interact. So the curcumin has a binding site in the ligand binding domain of the receptor, has a site for curcumin interaction. So that we had proven. And so typically what happens, that if you use curcumin, then you can see here the hormone receptor complex normally binds to the gene and do the trans-activation of anti-inflammatory genes or trans-repression of inflammatory genes. However, in presence of curcumin, you can see here the impact is much better, and so you can use less of dexamethasone or triamcinolone, less of glucocorticoid with curcumin, and that will reduce the side effect. So we try to see capsaicin. So we had purified capsaicin from US, and that we used, and it has typically gave the effect. So in curcumin had 100 micromolar. This had little more. Capsaicin, which is active ingredient of chilies, it requires 150 to activate the receptor. And this also has a binding site in the hormone binding domain of the receptor and give the effect. And so that we summarized exactly in the same mechanism. And this we had sent. This was my DST project long ago, and you won't believe on the front page of the highlights of extramural grant by DST in 2018, I think. Yeah, this was published. You can see here this is the receptor, and the yellow is the curcumin, and red one is the chili. The green one is the chili, and this had an interaction truly in the ligand binding domain, so that we moved ahead. So coming to the free radical, today one of the theories of aging is that we have more free radical generation during aging, and if we can quench the free radicals by any mechanism, whatever expertise you have, then you can have a better healthy aging. So as I said, protein oxidation is by carbonylation, that these protein carbonyls are produced. As I said, that particularly the arginine, lysine, and proline, these are the ones which are prone to carbonylation, where amino group is converted to carbonyl group. This you can see here for students that carbonyl group is generated. Now, there are many diseases which are associated with carbonylation, increased carbonylation of proteins, which is oxidative damage in the protein. And you can see here Alzheimer's, chronic lung phenotype, diabetes, and so on and so forth. So curcumin we used, and you can see here the protein carbonylation. See here, there is a young mouse and there is the old mouse. See how protein oxidation has gone up during old age. And when we used curcumin, this surprisingly, we published in this good paper, curcumin is not water soluble. So we had to dissolve in DMSO and dimethyl oxide. The DMSO itself has an antioxidative role. So you can see with DMSO in curcumin in DMSO had reduced the protein carbonyl content. And this, fortunately, I was in Japan at that time, so I bought some, earlier young student will not know, we used to do research by begging virtually, by borrowing things from abroad. I remember micropipettes were not there at that time. We used to do by mouth. So when you go to US, you ask your mentor that, could you please give me these micropipettes? I'll take it to India. So this the Japanese group had made first time the anti-carbonyl antibody, and that we took and bought it back. And you can see here there's the immunoblot where you can see the normal and is a control and there's the treated. So see that in young animals the protein carbonyl level comes down using curcumin. This is in the old case. And this about reactive oxygen species. Why you had more carbonyl content? So the question was in our mind. So we measured the ROS level in young and old in brain. And you can see here the old brain had this level of ROS. And of course, curcumin had reduced the ROS level. So the curcumin and capsaicin, we summarized till that time, curcumin and capsaicin both have a healthy aging potential by reducing inflammation and oxidative stress. And this is by potentiating the glucocorticoid receptor function by the curcumin and capsaicin as anti-inflammatory. And then we move to the later part. Bajradanti, you might have heard, which is Potentilla fulgens. And the root extract of Potentilla fulgens had excellent potential of antioxidative role. And that we studied with, look at the MDA, that malondihyde lipid peroxidation. And you can see here, this then liver and this then kidney. In both the cases, you can see here the young malondihyde and old one. Huge amount of lipid peroxidation in old age. But if you use the extract, you can see here it's much more significantly reduced. And we took vitamin C as a control. So you can see here in both the liver and kidney, this we measure catalase. Catalase is enzyme, which is antioxidative enzyme, which converts hydrogen peroxide to water. And you can see here the level of catalase goes down as a function of age in both liver and kidney. However, the extract had elevated the level. So if you elevate the level of catalase, it gives you more antioxidative impact and give relief to the animal. This we did by Western blot analysis. We had good antibody against the catalase. You can see here young, old, much reduced, and then by extract, it is much elevated in both the tissues. This glutathione peroxidase is also an antioxidant enzyme. And look here how glutathione peroxidase level is reduced in old age, both the liver and kidney, and the extract had elevated the level. And this we confirmed by Western blot analysis and published. And then we summarized that antioxidant potential of Potentilla fulgens offers promising opportunity for protection against oxidative stress-mediated pathophysiological changes during aging. Then we try to see, one of the students attempted to have effect of this Bajradante on diabetic mouse. Diabetic animals exhibit the very close pathophysiological condition of aging because you have more cross-linking in diabetes, which is also hallmark of aging process. So what he did, measured the lipid peroxidation. You can see here it goes up in liver and kidney in diabetic model. So we had an excellent model of diabetes in the lab using the Alexan and streptozotocin induced model, so that are having a higher glucose level, and that we try to use as an experimental animal. And then we had they look at the extract on glutathione peroxidase, very similar condition we had on this enzyme, that during aging the enzyme is reduced However, the extracts elevated the level. So this about SOD, the superoxide dismutase enzyme, which is again you can see here is reduced during aging and elevated by the extract. And this is also the catalase activity, which I have explained. So using both the models, we have confirmed that level of catalase, which is reduced during aging, and that is how you have more oxidative load because antioxidative enzyme is reduced. So the extract had elevated and gave the relief. This is the one which was very surprising and that we could publish. NF-kappa B is a pro-inflammatory. And you can see here the level of NF-kappa B binding to DNA, and you can see here it's elevated. So the notion that you have more inflammation in old age because NF-kappa B binding to DNA is more in old age. So that gives you the more inflammation. And look here, the extract has reduced the NF-kappa binding to DNA. That means that it has a beneficial effect and having antioxidative property. So the Bajrandi potentilla fulgans increases antioxidant defense and reduces inflammation via NF-kappa B in diabetic mouse model. And I think last slide before I close, this we published in 21, summarizing the role of nutraceuticals in healthy aging. And we have put all these, including resveratrol. Do you know resveratrol? The students, tell me one fruit which has huge amount of resveratrol. Have you heard this word? Resveratrol from students? Resveratrol? No? Where do you get from? Grapes, right. Right. And in grapes, you have different types of grapes. That is what yesterday Professor Lakodia was telling. The grapes from here and grapes from Nagpur, grapes from somewhere, there might be compositional differences in the components. And you know that you have the greenish one and you have the dark one. Dark one has more amount of resveratrol and it is excellent antioxidant, anti-inflammatory agent. So anyway, these are the mechanistic component. You need not to worry at that all these molecular parameters by whether it is resveratrol or ascorbic acid, that vitamin C, vitamin E, you have tocopherol, the curcumin, all these nutraceuticals, they have been used in our society. They might be authenticated more scientifically, but they definitely provide you the healthy aging potential. Thank you very much for your patience hearing. And these are my contribution of all my students, which I had the galaxy of students. They are all different places, and my foreign collaborators whom I had collaborated during my career. And I thank each one of you for this opportunity. I can pick up a couple of questions if some of you have. Please.
Okay, thank you very much, sir. It's a very nice lecture. I'm privileged to hear this lecture. So my question is that you have checked certain herbal herbal things or plant extracts on the diabetic mice.
Right.
So you found that the certain, like you have checked catalase, SOD, GLP and all these things that get reduced, the ROS level get reduced after using those herbal extracts.
Right.
Have you checked the blood glucose level of those diabetic mice? Because sometimes we found that the level of these enzymes get reduced. It's true,
Right.
but there's no effect in case of the blood glucose level.
That's
so
right.
Yes, sir.
Yeah, I mean the experimental design was such that the measure of these enzymes till the animals were diabetic. But you have a point. That's very important to see what happens to the animal to a particular duration. During that duration, you are right that the blood glucose level might come down, might not.
Yes, sir.
Yeah. Okay. Thank you.
Thank you, sir.
Well, those
Exactly dose is reached to the liver or kidney.
well, the pharmacokinetics of these drugs we haven't studied, to be frank. And the dose was, I think, the 100 micromolar that, I mean, after having a dried powder, the student had used. And the definitely we have not looked into the pharmacokinetics of the extract. Thank you. Oh,
Sir, thank you, sir. Excellent presentation. I have one question.
please.
Please give your opinion on that. Actually, oxygen is very essential for our life, for generation of energy and everything. On the other hand, oxygen itself generates reactor ROs and degrades lipids and proteins in our body.
Right.
At the same time, why nature it select oxygen as such a crucial molecule?
Just little clarity. Why nature selected
Oxygen for essential to our life. At the same time, it is a negative to our body also. Your comment only,
so
sir.
Nice of you. He asked the question which is very close to my chest for almost 35, 40 years. We used to call, please take your seat. We used to call oxygen as a dangerous friend. And the reason being that oxygen is essential. Can any student tell me what is the last use of oxygen in our body? Oh, this side. I want it from this side. Chalo bolo. Suppose you respire, you take oxygen, you expire, you remove carbon dioxide. When you inspire, you get oxygen. It goes through blood, hemoglobin, goes to tissues, go inside tissues, inside cell. In cell, where it is go, where it is utilized. Any student. Chalo, mitochondria, within mitochondria, in electron transport chain. Right. Says last stage of ETC in mitochondria. Excellent. So you have one atom of oxygen and two hydrogen, H2O. So that's the strategy which nature chose, and during that process you have oxidative phosphorylation, ATP, so on and so forth. So that we will not bother. His question is that if this is the scheme, then why the oxygen, same oxygen, is converted to superoxides, O2 with electron, then creating superoxide, then hydrogen peroxide, and blah and blah. So the answer, can any one of you tell why nature chose, reply to his question? Why nature chose to have superoxides or hydrogen peroxide? They are there. And why they are there? Okay, in other words, let me simplify a little bit. Tell me any good role of, good role of oxidants in the body. Every time we abuse reactive oxygen species, ROS is bad, ROS is bad, ROS is bad. Reactive oxygen species is bad. Can anyone tell me a good role of reactive oxygen species? Because nature will not do anything which is bad to us. The only that as we age, to be frank with you, aging should not be afraid of. Except, as I said, it is a part of our lifespan. Nature, I'm not talking about science part now, philosophy even. The nature chose all species to live at certain time. You cannot, if you look at the evolution, if somebody who is born will remain there, then what will happen to population dynamics, population control? So the nature, in order to even age, nature has these tools, provided these tools that after reproductive maturity, when you have done the nature's call, then slowly and slowly you get deteriorated and you are out. Our aim, if it is a natural process, then why do you study aging? The purpose is that if we know the mechanism how we age, then we can, as I said in the beginning, that purpose is not to live us forever. Purpose is to, as long as we live with good health, so that nobody is bothered to take care of us. So we can take care of our own business. So the nature gave reactive oxygen species for a purpose. I'm sorry, I don't have that figure here. ROS, that reactive oxygen species, has a strip. The pH, pH strip, low pH and high pH. ROS also has a strip. You have high ROS, you have low RS, you have in the middle. The middle one is the one which is there in our body to take care of system. If you take antioxidant capsules, that is how a lot of time the companies come up with antioxidant capsules, and there are people who have done it and which is not good. Don't take antioxidant capsule too much so that your ROS goes below the level. Because ROS, the reactive oxygen species, is also important. Tell me where ROS is involved. Huh? No, that's fine. But what I'm asking you that, is there any role of, this is a good science, virtually all students should keep in mind, because a lot of time we have a notion that, oh, ROS is bad, remove ROS, throw ROS, like cholesterol. Cholesterol, every time we say cholesterol is very bad, take cholesterol minus diet. But cholesterol is also essential. All your steroid hormones are synthesized from cholesterol as a precursor. Same way, during Corona time also, you might have seen, as a student, you must have studied immune system. What is the soldier in immune system? Who fights?
Sir, antibodies.
No,
Antibody.
Antibody reacts to antigen, neutralizes, that's fine. But if you look at the biochemistry of immune system, it is all pro-oxidants. These superoxides, the hydrogen peroxide, they are very good soldiers. So if you reduce your ROS to 0, you will die because of immune system. You have compromised your immune system. So one is, the ROS is necessary for fighting against the infections. 1. 2, cell division. Cell proliferation requires some amount of reactive oxygen species. And those who study very well, go into deep, you can search also, you'll find. So that's essential. And if you lower your oxidative load too much, then it is not good for your health. That is how nature chose, coming back to his query, that nature chose some of the oxygen to escape as superoxide to protect you from diseases and from having cell survival. So that's very important. You can search, you will get the answer. Thank you so much. Thank you so much for your patience here.
Thank you, sir. Thank you for gracing this event with your presence and sharing your invaluable insights. On behalf of everyone here, I would like to extend a heartfelt thank to our esteemed Professor, Dr. Ramesh Sharma, sir. And I now request SBTI President, Professor Edithil Vijayan, sir, to kindly felicitate our speaker, Professor Dr. Ramesh Sharma, sir. Thank you, sir, for such an insightful presentation. And the level of engagement during the Q&A session is a testament of the impact of your talk that we had on our students. The students were deeply involved and eager to learn and actively participate. Thank you again, sir, for your sharing your expertise and making this session so interactive and enriching. Now, further we'll move for technical session 3. Before that, there is an important announcement to be made. The remaining 2 award categories, the Kunut Pharmaceuticals Award for the best oral presentation paper presentation in herbal medicine and disease management, along with Professor Edithil Vijayan Award for best oral research paper presentation in neurosciences and neuroendocrinology, that we were left. Now we are going to take that, the 2 award paper presenters in room number 19 Ayurveda building. So all the award presenters are requested. The Kunut Pharmaceuticals Award and for Professor Edithil Vijayan Award, these presenters are requested to kindly head towards room number 19 Ayurveda building. And even the chairs and co-chairs of the sessions are requested to kindly proceed towards room number 19 Ayurveda building. Once again, I request all the presenters, the Kunut Pharmaceuticals and the Professor Edithil Vijayan Award presenters are requested to kindly head towards room number 19 Ayurveda building. And the sessions and the co-sessions for the chair and the co-chairs for the session are also requested to kindly proceed towards room number 19. The rest of the students can be here, and even those students who want to listen to those presenters can also move towards room number 19. Here in this auditorium, we are going to start with the 3rd technical session for today. We are just waiting for students to kindly settle down now. We are to begin technical session 3 now. Once again, very good morning to all our esteemed delegates, distinguished speakers, and participants. It is my honor to welcome you to the 3rd technical session of the 3-day international conference on biotechnological applications in Ayurveda and biomedical sciences, coupled with the annual meet of the Society for Biotechnologists, SBTI, India. As we navigate through an era defined by rapid technological advancements, biotechnology plays a pivotal role in shaping our future. This session will shed light on some groundbreaking developments in this space. To ensure the success of the session, we are privileged to have an outstanding chairperson and co-chairperson with us today. Before we begin, let me extend a warm welcome to the distinguished personalities leading this session. It is indeed a privilege to have with us Chairperson, Professor Dr. Sarita G. Bhatt Madam, and our co-chairperson, Mr. Sujit Nair, sir. So I kindly request the chairs and the co-chairs to kindly proceed towards the dais. We welcome you, sir and madam. Our chairperson for this session is Professor Dr. Sarita G. Bhatt, madam, who serves as the professor at the Department of Biotechnology, Cochin University of Science and Technology, with more than 25 years of teaching and research experience in biotechnology and microbiology. Ma'am's area of expertise include bacteriophage therapy, gut dysbiosis and health, gut microbiome, probiotics and its application, recombinant phage proteins as antimicrobials and plants. Ma'am has published more than 100 publications with high impact factor, awarded with several projects with worth a few crores of rupees. Recipient of UGC Mid-Career Award 2020. Ma'am, we are delighted to have your guidance leading this session. We request all the participants to kindly give a big round of applause for Madam. Joining Madam, we have with us Mr. Sujit Nair, sir, founder and technical head, SBM Pharma Technical, with a robust background in the pharmaceutical industry. Sir has brought over 20 years of expertise in analytical method development and validation, regulatory affairs, quality control, quality assurance, and quality management systems. Sir is dedicated to driving excellence in the field through education and training. We are fortunate to have Sir's expertise alongside Professor Dr. Sarita G. Bhatt, Madam. So kindly, please join me in welcoming a warm welcome to Professor Dr. Sarita G. Bhatt, Madam, and Mr. Sujit Nair, sir, as a chairperson and co-chairperson for the technical session 3. We have Dr. A.N. Singh, our faculty, biotechnology, as the reporter for the session. The session will feature insight lectures from eminent scientists and researchers in the field of biotechnology, biochemistry, and molecular biology. We are privileged to have with us today leading experts who will share their groundbreaking research, and I look forward to an engaging and enriching discussion. Thank you. So now we begin with our first presenter.
I would like to call Dr. Rupali Gupta ma'am and introduce her to the audience. Dr. Rupali Gupta is a distinguished researcher in plant microbe interaction at the Robert H. Smith Faculty of Agriculture, the Hebrew University of Jerusalem, Israel. Her research focuses on biochemical pathway in plant health, disease mechanism, and therapeutic advancement. Through her work, MAMS contributes significantly to microbial biochemistry and translational research in plant pathology. Today, MAMS will be delivering a lecture on the topic Microbial Modulation of Bacopa, a biosynthetic pathway and a defense mechanism in Bacopa monnieri under nematode stress. Let's warmly welcome Dr. Rupali Gupta ma'am.
Very good morning, everyone. It's okay? Okay. First of all, I would like to thank to the organizing committee for providing me this opportunity to discuss my work on this platform. And I'm going to discuss here my work on microbial modulation of bacoside, a biosynthetic pathway and defense mechanism in Bacopa monnieri under the nematode stress. So before going to the more detail about my work, first of all, I would like to discuss about that what is the impact of root knot nematode on medicinal and aromatic plants. So basically the plant parasitic nematodes are unsegmented, unciliated nematodes, which are endoparasitic in nature and causing over more than around 175 billion annual crop loss worldwide. And these nematodes are basically 3 types. either ectoparasites, endoparasites, or semi-endoparasites. So endoparasites are those which move around the plant root in soil and feeds on root ectoparasitically. Basically, the mouth part are inside the root and the other body part is outside. Endoparasitics are those which enter in the plant tissues and produce different kind of symptoms like stunting, slow growth, and yellowing of the leaf. And these nematodes are major economically important nematodes. Like different species of nematodes like Smelodogine, Heterodera, Rhodopholis are comes inside this group. And semi-endoparasites are those nematodes which partially entered into the root system, like the Rotilingus and Tilingus. So basically, these are the plant parasitic nematodes, which causes diseases in the plants. There are some free-living nematodes also. For an like example is C. elegans, which is a model organism and used for the study of aging and neurodegenerative disease. And as we know, last year there was a Nobel Prize on C. elegans. Can anyone tell me like on which study? The Nobel Prize on Physiology and Medicine, and it was on the model system was the C. elegans, and the prize was on microRNA, which controls the gene expression. So here I'm just giving the brief about what is the life cycle of root knot nematode and how it causes the disease on the plant's root. So first of all, the females nematodes lays eggs in egg masses on the root surface of the plants, and the first molt take place inside the eggs. And this releases the infected juvenile stage, and these juveniles invade the root tips and initiate the feeding site by the injection of esophageal glands and secretes into the root cells and forms the root galls. Here you can see the different galls patterns in the root surfaces, and this is the major symptom caused by the root knot nematode, which is the Melodogine species. And again, at maturity, females lays egg on root surface. So now the question comes, why we choose Bacopa monnieri as a plant system. Bacopa monnieri is one of the most honored plant, and it is a second in position in the medicinal and aromatic plants because of its presence of the bioactive constituent, which is bacosides and bacosaponins, which is useful in the many disease treatments such as the memory enhancer, anti-inflammatory, anti-diabetic. So our main major focus on this Bacopa monnieri, because of the growing demand of this plant and its bioactive constraint, it is necessary to grow these plants under the natural condition. That's why we choose this plant to protect the plants from the root knot nematodes and to enhance the bacoside egg content, which is the major constituent of this plant. And so that we apply the different, we focus on the microbial interventions like a biological management for the nematodes. So this is an overview of pharmacological effects of major bioactive constituent of Bacopa monnieri. Like I mentioned, bacosides, bacosaponins, and they are used in different for the different diseases treatment, like anti-cancer, anti-diabetic, prevent the neurodegenerative diseases, and for examples. So the disease caused by the nematodes causes different symptoms like stunting, chlorosis, leaf drop, yellowing, culling of the leaf, as I mentioned before, root galls formation, reduced growth, and irregular symptoms pattern. Before, the scientist was not very clear about these diseases are caused by the nematodes, because the symptoms we found get by the nematodes also found by the nutrient and the water disorders. So after the identification of these patterns like the gall formation, then they said, okay, this disease is caused by the nematode. There's a different nematode management techniques, like using the resistant plants. So farmers can grow the resistant plants in the field. Like one season they can grow the susceptible plants, and the other season they can grow the resistant plants. So the nematode population can decrease when they are growing the plants in the particular season. Organic abundance. soil solarization, crop rotations, following. Chemical nematicide is also one of the management technique, but it is banned in most of the countries because of its harmful effects on human and the environment. And the last is the biological treatment. Here we focus on the biological treatment because it's least harmful for the environment and the human. So now the question comes, why the kytonetic microbes? As I mentioned before, in the life cycle of the nematode, the female lays eggs, and these eggs are made of a chitin layer. So if we rupture the chitin layer with of the outside nematode eggs, so the premature hatching take place. So these the released J2 stage were not able to infect the plants. So our first research was to focus on the isolation, screening, and characterization of the kytonetic microbes from the Bacopa monnieri for the management of nematodes. So first of all, we isolated different bacteria using the colloidal chitin as a agar plate, and the colloidal chitin was here the substrate by utilizing by the chitinase producing bacteria. So here you can see the clear zone outside the bacterial growth, which shows that these bacteria are able to utilize the colloidal chitin as a carbon source and release the chitinases. This is the different morphology of the bacteria, which are chitonetic in chitinase producing. So first of all, we did the quantification of chitonetic activity of the selected isolates. And here we coded these different isolates as MTN to MTN24. And we found that, okay, all the bacteria, the 6 bacteria able to produce chitinase after 96 hours of incubation. We further check the plant growth promoting activities of this bacteria and biocontrol activities like ammonium production, hydrogen cyanide, protease, cellulase, pectinase, amylase, and lipase production, which is helpful in the plant growth promotion activities. This is the pictorial representation of the protease activity, gelatinase, and cellulase. Also, we check the phosphate solubilizing and the siderophore activity. The phosphate and the siderophore present in the soil is in insoluble forms. So basically, the plants cannot utilize that phosphate directly. So these microbes are helpful in solubilizing the phosphate and make it in a soluble form so that the plants can utilize those phosphate from the soil. And siderophore production is also like an iron chelating agent that have a high affinity to bind with the iron. So basically, the MTN14s have more phosphate solubilizing activity, followed by the MTN15, and the least was in MTN19. Same, the siderophore activity is most in the MTN18, and the followed by the MTN22, and the least was in MTN24. So this is the pictorial of phosphate solubilizing activity. Here you can see in the agar plates and in the liquid form, and also the siderophore production. So we also screen these bacteria for the plant hormones like IAA production, which is indole acetic acid, and gibberellic acid production, and which is maximum in the MTN14 and followed by the MTN22. So our main focus on the biocontrol activities of these bacteria, which also could have a potent nematicidal activity. So to further confirm this activity, we did the nematicidal we checked the nematicidal activity against the egg. So against the plants, the nematode eggs and the J2 mortality, and we found maximum in the case of MTN18 and MTN22. So here you can see the live nematode picture, and this is the dead nematodes. And we identified these bacteria on the basis of the phylogenetic gene and also the Chi-A gene, which is responsible for the chitinase production, which further confirmed that okay, these genes are present in these bacteria. And we identified these bacteria and found that MTN14 is, MTN14 is Streptomycetes, MTN18 is Streptomycetes albus, like MTN16 is the Microbacterium, MTN22 is the Chitinophyllus. So till now, okay, we identified these bacteria, we isolate and found that these bacteria have nematocidal activity and the plant growth promoting activity. So we further confirm our result under the greenhouse condition using the natural and the sterilized soil. So in this experiment, we found that the Streptomycetes have more plant growth promoting activities, while the Chitinophyllus microbes have more biocontrol activity. So we thought that why not we combine this bacteria and apply it in a combination. So in our next experiment, we checked the compatibility test between these two bacteria, that these bacteria are synergistic to each other, not antagonistic. And further, we applied these bacteria in a single treatment like T1 to T4. Here you can see that these are the cellular bacterium, Streptomycetes, Flavobacterium, Chitinophyllus, and the T5 to T10 is the dual microbial treatment. And it was found maximum in the case of the T9 treatment, which was we expected the most biomass yield and the reduced Gallon index, and the total bacoside content in the percentage is also higher. And also we compare our result with the chemical nematicide, which is carbofuran. So after that, we further check these bacteria for the plant growth promoting activity, and we check different defense-related enzymes in plants, and like total phenolic content, total flavonoid content, free radical scavenging activities, and the total antioxidant capacity. So from the greenhouse experiment under the sliced and natural soil condition, we found that these bacteria are able to manage the disease and increase the biomass production of the Bacopa monnieri. So further, we try to evaluate or identify that how these bacteria are able to reducing the nematode population in the soil. So for this, we evaluate these bacteria and found that these are able to mitigate the oxidative stress. Before the previous presentation by the Professor Sharma sir, he already explained about the reactive oxygen species. And to mitigate the when the nematode infect the plants, they generate the oxidative stress. So if you can manage this oxidative stress by the production of the various antioxidant enzymes, so that is responsible for the plant growth and managing the disease. So in this experiment, you can here see that there is a very less biomass production in the control with the nematode infection, while the maximum in the dual microbial treatment with and without nematode. So from our previous experiment, we found that, okay, these bacteria are able to increase the biomass production and also increase the total bacoside contents, and which is as expected in the dual microbial treatment with and without nematode. And the maximum least bacoside content and the least biomass production was in the control with the nematode treatments. We further confirmed by the FTNIR spectra analysis about the bacoside content, and it was found in the microbial combination and the least nematode population and reproduction factor and root gall index in the control plant with nematode stress. So this is the diagram, histochemical analysis of the ROS production. So we checked the ROS production on the Bacopa monnieri leaf, and we found maximum in the control treatment with the nematode stress, while the least in the dual microbial treatment without Meladogine incognita, the root pathogen, and also in the with nematode stress in the dual microbial treatment. And this is the histochemical analysis of the H2O2 production, which we identified by the DAB staining. And it is maximum, as you can see here, the maximum H2O2 production in the control with nematode stress and the least in the microbial treatment. Further, we confirm our result with the superoxide radical by the NBT's chemical, and it found maximum in the nematode stress treatment. And this is the PCT program cell that which we did by the advanced blue staining, and it is one of the parameter of the plant pathogen interaction, and it's found maximum in the nematode plus control plus nematode stress, and the least in the dual microbial treatment. So further, we confirm our result by using the SEM staining, like what is going on with the root reason. And we confirm our result with the SEM staining. And as you can see here, there is a more root damage in the nematode stress. And also there is egg masses in the microbial treatment with and without nematode stress. Here you can see the colonization, and the root structure is proper as much better than the control plants. So this is the mechanistic representation depicting the mode of action adopted by these two bacteria. And so when we applied these bacteria in the root region, also the nematode, it increased the development of the disease symptoms increase, and it's also mitigate the oxidative stress in the terms of H2O2 production, singlet oxygen, and these bacteria able to mitigate the oxidative stress that is generated by the nematode pathogen. So till now, okay, we found that these bacteria are able to mitigate the oxidative stress to manage the infection, that is infestation caused by the nematode. So we further evaluate like how the bacoside content is increasing in the plants that is treated by the microbes. So for this, we further did the experiment in the greenhouse condition, and in the picture you can see here the least biomass production in the nematode treatment, and the maximum there is also flowering of bacoside, Bacopa monnieri, and in the dual microbial treatment. And this result further confirmed that, okay, there is a more biomass production and a less nematode population and root galling index. And this is the HPLC analysis of bacoside content, and it's found maximum in the treatment of microbial treatments, and the maximum in the case of dual microbial treatments. So this is the bacoside A biosynthetic pathway. As you can see here, there is this is well known. Okay, there is a two pathway, MVA pathway and MEP pathway. It is in cytoplasm, and this is in the plasmid, which is further responsible for the production of the triterpenoid saponins. So basically, acetyl coenzymes acetyl coenzyme is converted into IPP, which is isopentyl diphosphate, via the HMG coenzyme A, reductase, and mevalonic acid. And this further, the whole process transferred the carbons for the production of the saponins. And we choose the key enzymes for this. And this is the HMGR, which is 3-hydroxy-3-methyl-butyl coenzyme A reductase, mevalonate diphosphate dicarboxylase, and this is the succulent synthase, that is key enzymes for the bacoside A biosynthesis pathway. And this result further confirmed there is a more gene expression when we applied these bacteria for the increasing the bacoside content. And also we check the defense gene, which is PR1 gene. This is defense, the pathogen related gene, and it's more in when we apply the nematode populations. We further check the various defense related enzymes, and it's in the terms of the total phenolic content, flavonoids, PAL, which is phenyl ammonium ligase, and total phenolic oxidate, and polyphenol oxidase. So phenolic is also one of the indicator for the plant pathogen interaction, like gallic acid, syringic acid, and ferulic acid have antimicrobial properties. So we found that, okay, these phenolic productions are more in the microbial treatment, and some are phenolics are not detected in the nematode stress. This is the when we applied these bacteria, we found that these bacteria also able to increase the cell wall strengthening in the terms of the lignification. And in this picture, you can easily see that the lignin is by the blue ring structure, and it's very less very less in the nematode stress, while the in microbial treatment, the lignification is very strong. It shows that these bacteria are able to enhance, able to manage the cell wall strengthening. Also, we check the callose deposition, and here you can see the blue-green fluorescence, which shows the callose deposition, and it's maximum in the microbial treatment, while the least in the nematode stress. So this is the mechanistic figure representing the chitolytic bacteria mediated secondary metabolites enhanced, like bacoside A content, which is the active constituent of the Bacopa monnieri, and defense responses in Bacopa monnieri. And so here, when we applied these bacteria in a root region, also the meladogide population, it's release the signals which received by the receptors of plant tissues. And these signals are strengthening the mechanical barriers in the terms of the callose deposition, lignification, activate the phenylpropanide pathway like phenolic acids, and also increase the gene expression of the bacosides A biosynthetic pathway. So till now, we validate our isolated bacteria in the greenhouse and field condition. So now the question comes, like, are these bacteria are also have a strong potential in the field condition? Because in the greenhouse condition, we have a standard temperature, humidity, but in natural condition, we have different types of factors. So we validated these bacteria under the nematode stress infestation field. So for this, we applied the bacteria and checked the bacoside cultivations, and after the 45 and 90 days of Bacopa monnieri under the nematode infested field. And here you can see the clear patches in the field, which shows that there is a nematode infestation. And further, we check the biomass production and the root knot index, and the biocontrol efficiency is more in the dual microbial treatment, like Chitinophyllus and Streptomycetes. As I mentioned before, like Chitinophyllus have more chitinase producing capability, while the Streptomycetes have plant growth promoting activity. So we further confirm the bacoside A content, and which is maximum in the microbial treatment. So, but in field condition, how these bacteria are able to manage the nematode population as well as increasing the bacoside content of the Bacopa monnieri. Yes. Okay, ma'am. I'm just going to finish. So for that, we try to analyze that, okay, when we applying these bacteria as a soil range in the root region, are they also modifying or shifting the microbial communities and making the plants with the root colonization with more beneficial bacteria? So for that, we check the chitinase production in the soil region by using this technique, and we found there is a more chitinase activity in the soil in microbial treatment. And the population of 16S RNA gene, which is phylogenetic, and the Chi-A gene is the more in the copy number, as we expected, because there is a dual microbial treatment. So the more bacterial population as well as most chitinase activity. And this is the DGG pattern, which is used for the metagenomic analysis, which is gradient gel electrophoresis. And in this, each band represent the microbial community. So you can see here that the lane 4 have more number of bands, which shows that there is a more microbial population in the when the plants is treated with the microbes. And like here, there is a dendrogram which represent there is a different microbial community in the dual microbial treatment, followed by the different types of community in the single inoculations, and the control is totally different from the others microbial treatments. And this is the mechanistic figure of the field elevation of these microbes. And here we applied the bacteria in the root region. It's able to enhance secondary metabolite content, which we further validated by the bacoside per A production. And in the soil system, these bacteria are able to changing in the structure and the function of the microbial community, which is validated by the copy or number genes.
Dr. Gupta.
Yes, ma'am, it's almost finished. So this is the conclusion. These bacteria have more chitinase and nematicidal activity validated in the root conditions, also manage the oxidative stress, and also cell wall strengthening and modulation of the bacoside A pathway, and activating the systemic defense in the terms of the accumulation of phenolic acids, high abundance of the Chi gene, and the shifting in the microbial community, which is further validated by the PCR-DGG technique. And thank you so much.
Thank you so much, Dr. Gupta, for a very excellent presentation. Since we are running out of time, I can take just 2 questions from the audience.
Yes. Yeah, it's different. Here we focus on the root knot nematode, because the symptom is totally different from the other plant nematodes. Sorry. Yeah, sorry. Can you hear? Okay. Actually, here we focus on the root knot nematodes, and it is the symptom is totally different from the other root knot nematodes, like cyst nematodes. There is a cyst formation, and the root knot nematodes, there is a knot formation on the root surfaces. So you get easily identified by the formations of gall. And the melodogyne species is different species of melodogyne, like melodogyne incognita, javanica, arenaria. Those are making the knots on the roots. And this is most economically important pathogen. That's why we focus on the melodogyne species, and also the Bacopa monnieri, which is major important plant and comes in the 2nd position in the Ayurveda for its bioactive constituent, which is helpful in the various diseases.
Any other question? That is 2 questions, so we will be finished with that.
Yeah. It's depend upon what kind of organic material we are using. People are also using aromatics and medicinal aromatic plants waste materials. After extracting the oils, which is also have medicinal properties, like you can take an example for the mentha. After extracting the essential oil, the mentha waste can be
Dr.
applied
Gupta?
in the nematode infestation field.
Yeah.
Yes.
So we will have a further discussion during the tea break. Now we'll move on to the next presentation. The compare will introduce the next speaker. Thank you so much.
Thank you so much.
Thank you so much, ma'am, for your insightful presentation. I would like to welcome our next speaker, Professor Dr. Yamini Bhushan Tripathi, sir. I would like to introduce sir to our audience. Sir is a renowned expert in medicinal chemistry at Banaras Hindu University, India, with extensive research in experimental medicine, phytopharmaceuticals, and metabolic disorders. Sir has made significant contribution to the field. His work, including patented innovations and numerous publications, has advanced medicinal chemistry and therapeutic drug development. Today, we have Sir delivering a lecture on the topic Integration of Science and Ayurveda towards Management of Metabolic Syndrome. Let's warm welcome Professor Dr. Yamini Bhushan Tripathi, sir.
Thank you to the organizers for inviting me. My special thanks to Dr. Prerna Aditi, the head department of biochemistry. She contacted me and invited. And I'm really happy to have a chair and co-chair, which are very much related to Ayurveda. Because in Ayurveda, we always think they are all scientists. One is the biotechnologist, right? And one is the pharmacy. But in Ayurveda, you know that these technologies are really required to validate our concepts and our products. With this introduction, how many minutes I have? 35 minutes? Okay. Okay. I dedicate my talk to my father. He was also the Dean Faculty of Ayurveda in Banaras Hind University. And he used to say in those days when I was in 1977, when nobody was interested in doing work in Ayurveda, no basic scientists, everybody was saying, no, don't spoil your life by working as a biochemist in Ayurveda. PhD, I am basically a biochemist. He used to say that Ayurveda is a science and it can only be proved at the molecular level. The best laboratories of the world can do science, then only Ayurveda can be put. And today it has come true. You see, all the 3 Nobel Prizes from 2015, 2016, and 2017, they have all gone to the concepts which are already being practiced by Ayurvedic people, but not scientifically validated. These people did. You know, the 2015 Nobel went to the lady who said that the extract of Artemisia annua is effective in killing the resistant bacterial malarial parasite. Because artemisinin is a molecule which was not effective. But when she used the extract, it was effective. It means the extract has a multi-targeted action, and synergistic effect of the extract is having some signaling pathway which is doing better action. Of course, the concentration of the active molecule may be less, but the therapeutic response is very high. And you know, if you go to the 2016, it went to the autophagy. There's a Langharam Maha-Aushadhi, as an intermittent fasting. I will come to this slide. And the last slide is the sleep. What is the melatonin cycle, how the sleep cycle is controlled. They have told the mechanism, but we were always telling about these 3 things from since centuries. And the most important is the extract is more effective. I have done the bioinformatics. What my previous speaker said, that the earlier wet lab was first. We have to do all animal experiments, then we go to the bioinformatics. But now it has reversed. Bioinformatics is first and wet lab is second. पहले हम लोग कंप्यूटर पे खोज लेते हैं, तब हम लोग रिसर्च स्टार्ट करते हैं. This is the science I have shown. This is I have done the hub gene found for the neural degeneration in a particular plant. You say there are more than 50 phytochemicals and genes which were involved, and 4 phytochemicals. They are acting on the hub gene. What I mean to say that by using the one extract, we can target many signaling pathways and many pathways. And this is very much similar to Ayurvedic concept of Vata, Pitta, and Kapha, which will come to the next slide. So validation of disease pathogenesis through bioinformatics. You know, the Vata has in Ayurveda, Vata, Pitta, and Kapha. The Vata has 80 diseases. Pitta has the 40 diseases. And the Kapha has the 20 diseases. But in modern medicine, all the diseases have been given specific name and the specific line of treatment. That is the problem with the allopathy and Ayurveda. Because एक ही साधे सब सधे, सब साधे सब जाए. So Ayurvedic physician says that if you treat the Vata, you are going to treat the 80 diseases. But here we are saying, like for diabetes, endocrinologists, for heart disease, cardiologists, for every disease you have expert. For WHO has set a guideline for the treatment. But here we say only treat this 3 things. A second thing I want to feel that okay, this Vata I am going to focus because it is the Prana Vayu. There are 5 types of Vata. It means that there should be 5 kind of diseases also. There should be 5 kind of hub genes also. The gastrointestinal disease, sensory disease, cardiovascular disease, musculoskeletal disease, and neurological disease. So all these diseases are governed by Vata, and but Vata are 5 types. So I think there is a need to do research how these hub genes are these systemic metabolic diseases are having this kind of hub genes, but how they are connected with these 5 Vata. Nobody has worked on it. This is the time for the scientists to work on it and how we can work. So this is the second Nobel Prize. I told you that the science behind the religious practices. Every religion has a fasting. Recently, that Ramjan was there, 1 month fasting. In our tradition, Ekadasi Brat, Mangal Brat, Brihaspati Brat, even every religion, it has been given what kind of fasting you should do. It is not only that you should not eat anything. There is a ras ahar dependent, there is a fruit dependent, there is a salt depletion dependent. तो हर प्रकार के हमारे यहाँ रिलीजन्स में, हिंदू रिलीजन में डिस्क्राइब किया हुआ है कि कौन सी फास्टिंग करेंगे तो क्या लाभ होगा. But this is very much a scientific, because in Ayurveda they have described there are 7 types of langan. That if one langan is you have to depend only on water. That is the most difficult, and then all the langans are easier. So this is what is the science behind it. That okay, okay. Here is the autophagy. You know that he said that the forceful restriction of diet enhances autophagy. There is a pathway mTOR dependent. mTOR regulates autophagy, and our fasting is taking care of that. So today in the topic I am going to cover 5 topics. The basics of Ayurveda, concept of health and diseases in Ayurveda, metabolic syndrome through window of Ayurveda and allopathy, and Ayurvedic as a supportive medicine, because I am going to integrate Ayurvedic medicine and allopathy. So, but I am going to talk only of science part, not the medicine part. And Ayurveda is not only the medicine, it is also important to know. When you talk of Ayurveda, people say that it is herbal, or it is something, some medicine has to be given. But in allopathy, medicine is the only treatment. In Ayurveda, your lifestyle, your diet, and your medicine, all the 3 things. So when you are talking of Ayurveda, you talk all the 3 things together, not only herbal medicine. And last point, some examples of research from my laboratory. So this is obesity. I'm going to talk more about it, because obesity is the cause of hypertension, sleep disorder, all the diseases are linked to this. But why it is happening? Because it is related to chronic kidney disease, CVD, fatty liver, type 2 diabetes, and cancer also. So this is the metabolic pathway for obesity. Everybody knows that main thing is I have told you, this is the hyperinsulinemia, glucose intolerance, lipid abnormality, and dysfunction of nitric oxide. So these are the mechanisms. A combined effect, metabolic syndrome is defined as a combination of obesity, blood pressure, diabetes, and hyperlipidemia. Ayurvedic says hypertension is a Rakt-gat Vat, dyslipidemia is a Medo Rog, diabetes is the Prameh, central obesity is the Sthol. You know, in the diabetes, Ayurveda says there are 20 types of diabetes. In modern medicine, we say 2 types of diabetes. One is the insulin dependent, another is non-dependent. But in Ayurveda, it has given the word Prameh. There are Kafach Prameh, Vataj Prameh, Pittaj Prameh, and Vataj Prameh. And Vataj Prameh is insulin dependent, like this is incurable. It cannot be cured. So when you are in that case, that is called the diabetes. So, but the other 2 prameha can be prevented. It can be delayed. So major pathways involved in the metabolic syndrome, as you know, that insulin signaling pathway, adipokine pathway, AMPK pathway, mTOR pathway, NF-kappa B, what our previous speaker, Dr. Sharma, told in great detail, and this, and toll-like receptors. And this line of the treatment in metabolic syndrome, the basic principle, what we correct the Agni, balance doshas, and reduce the Meda Dhatu in the obesity. And here symptoms wise management of all the 4 concepts. This is the main difference. What is Agni? When we come to the next slide, that your digestive power is very important in your GIT and in all the cell. The catabolic and anabolic pathway balance is important. That is called the Agni. And the lifestyle modification, Ahar, Nidra, and Brahmacharya. There are 3 pillars for good health: food, sleep, and code of conduct. Dincharya, Ritucharya, and Kal Bhojanam. Dincharya means circadian rhythm. Ritucharya means seasonal rhythm. And Kal Bhojanam means time of eating. These are the basis of Ayurveda for control the obesity. The Pathya is the high fiber diet. This is similar to modern medicine. Herbal medicine is there are 6 rasa. I will come to the next slide and I will explain. Then Panchakarma. Panchakarma is the removal of the toxins from your body. And then is the yoga and pranayam. This is the exercise related thing. And the targeted action. If after these things, then you start the medicine. That is the targeted action. So what happens in obesity? You know, your adipose cell becomes inflammatory. जो फैट है हमारे में, फैट बहुत जरूरी है, जैसा डॉक्टर साहब ने बताया कि फ्री रेडिकल बहुत जरूरी है, लेकिन ज्यादा हो जाएगा तो गड़बड़ है. आपके यहाँ जो आपका जो मोटापा है थोड़ा होना चाहिए, नहीं होगा तो बॉडी एकदम हवा की तरह हो जाएगी, एजिंग बहुत जल्दी हो जाएगा. लेकिन ज्यादा फैट हो जाएगा तो गड़बड़ हो जाएगा. तो ज्यादा फैट मतलब होता क्या है? They become inflammatory. The macrophages, there are M1 is the inflammatory and M2 is the your native immunological regulator. तो macrophage are the best first line of defense in your body. लेकिन अगर macrophage घुस गया है adipose site में, ये inflammation produce कर रहा है. देखिए जितने inflammatory cytokines हैं सब produce हो रहा है. तो we want good fat, not the inflammatory fat. This is very important. अब मैं आयुर्वेद की तरफ आ रहा हूँ. तो concept of योग, there are 5 koshas, you know, अन्नमय कोश, प्राणमय कोश, मनोमय कोश, विज्ञानमय कोश, and आनंदमय कोश. And there are 7 chakras, you know, मूलाधार, sacral chakra, स्वादिष्ठान, मणिपुर, अनाहत चक्र, विशुद्धा चक्र, आज्ञा चक्र, and सहस्त्र चक्र. And these things are related to your naturopathy method, detoxification, pranayam, hypertrophy, stress management, this meditation, and right understanding of health and lifestyle. This again, Anandamaya Kosh is the complete meditation. So what I mean to say that your yoga and science are very much connected, but not much work has been done. We have to work on that. You see, these chakras are related to some glands, or some papers are there in the already PubMed. There's a sense of the pineal gland, pituitary gland, thyroid gland, and the thymus. Pancreas is the Manipur chakra, and the gonads is the Swadhisthana chakra, and the adrenal gland. So these are the, you know what happens for chakra maintenance is the real pathway to control your sympathetic nervous system and parasympathetic nervous system. Parasympathetic nervous system is relaxed stage, and sympathetic nervous system is stressful stage. If you are having a lot of stress in your body, then you are running here and there. You are always stressful to do some work. Anxiety is there. That is causing sympathetic nervous system, which is not good. Parasympathetic, when you go to the meditation, you know, in yoga there are the 7 Ashtanga yoga: Yam, Niyam, Asan, Pranayam, Pratyahar, Dharana, Dhyan, Samadhi. The 4 last one is that parasympathetic nervous system stimulant. Do like that. And this is the, I want to introduce Ayurveda. There are 8 branches, and there are 3. What is the objective of Ayurveda? Swasthasya swasthya rakshanam, atrasya vikas prasannam cha. Means a healthy man should remain healthy, and the diseased person should be treated. For this, they made the 8 branches, Ashtanga Ayurveda. 3 Vajikaran, Rasayan, and Bhut Vidya. They are the 3 established branches of Ayurveda. They are the mainly meant for swasthasya swasthya rakshanam, a health for maintain the wellness of a healthy person. And other 5 are for the treatment. So what was you see that 90% people are healthy in the society. They're not going to the doctors. Then हम लोगों को डॉक्टर्स को सोसाइटी में जाना पड़ेगा, जो आउटरीच प्रोग्राम करते हैं, उनका स्क्रीनिंग करके उनको इसके बारे में हमको बताना पड़ेगा. क्या बताना पड़ेगा? Sorry. These are the 3 major books I wanted to say: Charak Samhita, Sushruta, and Vagbhat, Ashtanga Hridaya. These are the major books. बताना ये पड़ेगा कि हम लोग हैं क्या? What is the life? The combination of the body, sense, organs, मन, psyche, and soul. ये तीनों का जो बैलेंस है वही आपकी लाइफ है. और ये जो पंच महाभूत हैं, इससे से वात पित्त कफ बना है, इसी से सत्व स्तंभ बना है. I'm coming to the next slide. This is a very interesting slide. I made it. You see, the Panch Mahabhutas are making your body, and Panch Mahabhutas are making your food, your medicine also. And what is the combination? What is the rationale of this? What is the science behind it? You see, Jal and Prithvi, they are making the Kaft. And Jal and Prithvi is making the Tamsik Guna. Saturajantam Tamsik. And Jal and Prithvi is making the Madhurahar. So the man who is having more Tamsik Pravitti, more Kafaj personality, should not eat much of the Madhuras. This is the science. Based on this, only Vata, Pitta, and Kaf and the Dravigun, Rasgun, Vir, Vipak has been designed. And people are using it for a long time. But science has not been done. So, okay. So basic concepts of wellness, there I told you earlier, Ahar, Nidra, and Brahmacharya, the three things. Cause of disease is also important. They also, Ayurveda says, remove the cause of the disease and then treat it. Only giving the medicine will not work. Lifestyle change and the cause of the disease has to be removed. I will come to the cause. What are three causes? Excess use of your sense organs, all the 5 sense organs. If you're excess using, you are causing a disease. And chronobiological, the seasonal change diseases. For example, in January, February, you get allergen, so asthma is there. In the rainy season, you get diarrhea is there. So these are the seasonal changes you have to manage. And last is the prajnaparat, psychological insecurity. 66% of the diseases are psychological insecurity. What is prajnaparat? Well, I tell to the class of the Ayurveda when I teach that मन और आत्मा के द्वंद्व को प्रज्ञापरात कहते हैं. मन क्या है? चंचल है, हमेशा क्या गुणा भाग करता है, क्या सही है क्या गलत है. आत्मा तो भगवान का अंश है. It has come from the God. So मन और आत्मा always tells the truth. What is called the gut feeling. Don't do it. But your man says, okay, let us take a chance. So that is called the prajnaparat. If you are doing wrong things knowingly, is prajnaparat. Today or tomorrow, it is something, some toxin is getting accumulated in your mind and your body, and this is being manifested as a disease. This is very important. And there are 3 lines of treatment in Ayurveda. Deva Vyapasya, Sattva Vajay, and Yukti Vyapasya. Deva Vyapasya means spiritual healing. Sattva Vajay means psychological counseling. And if these two don't work, then go for Yukti Vyapasya. What is Yukti Vyapasya? Lifestyle means sleep and diet, body purification, and then medicine, herbal, mineral, and animal food. In Ayurveda, there are 3 kinds of medicines. Yesterday, Dr. Lakhotia told about Ras Aushadhi, and today Dr. Sharma told about the lifestyle, sleep and diet is very important. And yoga, I told you, Yam Niyam. Yam Niyam is very important. You know what happened? In the whole world, yoga has become very popular. But a perception is that asana and pranayama is the yoga, which is not true. Yam Niyam is the heart of the Ashtanga Yoga. Means this is your personal hygiene and your social responsibility. This has to be covered in this. So with this word, I go ahead. So gap to synergize in Ayurveda biomedical research is the no common playground. The scientist doesn't understand Ayurveda, and Ayurvedic people doesn't believe in science. That has a playground. The football khelna hai, to playground to hona chahiye. So each discipline has to understand the terms of other. Then only we can talk for a collaboration. And both our experts need to understand and collaboration rather than doing job. Suppose I am a biochemistry. If I started doing the Ayurvedic clinical trial, that may not be correct. But a man who is doing clinical practice, he should contact with a biologist, biochemist, to give the sample and let them do it. So repetition of work is not good. And then therapeutic claims of Ayurveda are not straightforward. This is very important. In modern medicine, we know, as Dr. Sharma said, you have a receptor and you have a ligand. Receptor-ligand binding helps. But in Ayurveda, it can be indirect. I am getting Triphala. Triphala is may not be acting directly on the receptor of GIT, but some hormones are being modulated. That hormone is affecting the GIT movement. That has to be worked out. This is a very important part. So 1 + 1, 2, it cannot be true with Ayurveda. We have to think on this. So clinical evidences are there in Ayurveda and advanced biological tools. If these things are kept together, novel drugs and novel concepts can be created. So tips for the wellness, Ahar, Nidra, and Brahmacharya, I told you. So Ahar, I am going to talk more because Aharam Mahaushadhi has been told. So there are 6 rasa, Madhur, Lavan, Katu, Tikt, Kasai, Madhur, Aml, Lavan, and sweet and sour. These are the combinations. I'm not going to detail. We are with Ras, Guna, Veer, and Vipak. There are 4 characteristics of any medicinal plant. That has this known by the Ayurvedic people. We don't understand in this from scientific term, but how the yesterday she talked about the personalized medication. This is the personalized medication. So we study the Ras, Gun, Veer, Vipak, and then we study the Vat, Pitta, Kapha. Depending on that, we combine it. So then another important factor, the incompatible diet. There are 18 conditions when the same food it become incompatible for one person and become compatible for other person. That has to be worked out. There are rules of eating. Rules of eating are also important. You know, somebody in Ayurveda, and I think in South India also, people first take the sweet and then go for food, and last they again take the sweet. But here, you know, soup is first. Soup is the astringent. It may increase your hunger, but it is not good. But taking sweet gives you a satiety, so you eat less. So obesity will be less. Similarly, if you take a sweet in the end, the digestion will be better. So what I mean to say is that the method of eating is also given in Ayurveda. So there are 18 points which Vaidya considers while prescribing you what to eat and how to eat. And then this is the example of incompatible diet. Like, for example, the fish and milk should not be taken. Honey and ghee should not be taken together in equal amount. There are many examples you can see. And the rationale of this incompatibility is milk and banana having common allergen may aggravate asthma. Similarly, these are the things which I have taken from the Charak Samhita. I have translated it. So science behind the bad health taking incompatible diet. It increases the inflammation, oxidative stress, combination of food having digestive issue can be there, GIT imbalance, and the undigested starch get fermented, and then the microbiome, intestinal bacteria may get disturbed. To change the cellular pH. So with these words, I go to Kala Bhojanam, time of eating. And Ayurveda has given a very strong stress on time of eating. Suppose you are getting in the morning at 4 a.m., then your time of breakfast will be different. You are getting up in the 7 a.m., time of breakfast will be different. And there should be a gap between the 2 meals. 4 hours gap is minimum. Suppose you are continuously eating, your Agni is being compromised. So hunger, other important point, when you are hungry, then only you eat. If you are not hungry and eating, you are going to cause a disease. So this is the circadian rhythm I want to tell you. Vata, Pitta and Kapha cycle is there. From night 10 p.m. to 2 a.m. is the Pitta cycle. In this time, liver has the cleaning work. But if you are eating, the liver also has the digestion issue. So how the one organ can do both work? The work is compromised and you are getting a disease. That's called Amekmusan. And Vata, some people ask me, what is the best time for exercise? You know, Vata is from the 2 a.m. to 6 a.m. This time, and the exercise also increases Vata, you know. Vat बढ़ता है और इस समय भी वात बढ़ता है. तो जो असली समय जो कफ बढ़ता है, इस समय एक्सरसाइज करना चाहिए. 6 AM से 10 AM के बीच में एक्सरसाइज करेंगे. और हमारे यहाँ 2 ही बार खाना कहा गया है, ब्रेकफास्ट, लंच या डिनर नहीं कहा गया, लंच एंड डिनर. अगर आप ब्रेकफास्ट आप कर रहे हैं लाइक ए वेस्टर्न पीपल, then you have to have a light lunch. Otherwise, have a heavy lunch and light dinner. So this is the time of eating, Vata, Pitta, and Kapha cycle. So I have made a description what you should eat when and what you should do, lifestyle and food activity. I'm not going to detail. Circadian rhythm I have told you. So I am forced to focus. Okay, time. Oh, liver is the organ, you know, it has its own circadian rhythm. So circadian rhythm means उसका टाइम जो है, जैसे दिन में 10 से 2 खाने का टाइम है बेस्ट, और वही उसी समय खाना पचाने का टाइम वो लिवर करता है. और रात में 10 से 2 अगर आपने खाना खाया तो फूड जो क्लीनिंग का, बॉडी क्लीनिंग का काम लिवर का है. लिवर हैज 15-16 फंक्शंस, यू नो, सम आर फॉर क्लीनिंग, सम फॉर डाइजेशन. तो इसे डिवाइड करके रखा है. तो हर ऑर्गन का अपना सर्केडियन रिदम है, बॉडी का भी सर्केडियन रिदम है. वी हैव टू सिंक्रोनाइज. जैसे कहते हैं यत पिंडे तद ब्रह्मांडे. What Dr. Sharma said, the epigenetic factor is more important than genetic factor. Epigenetic means environment where you live. जैसी संगत है वैसा ही आपका वो हो जाएगा, personality हो जाएगी. Even the twins have a different kind of behavior because the environment is different for them. So another life is the thing is the use of the, this is important for the common people, rejuvenating food supplement, body purification, control of emotions by yoga, Ashtanga yoga, use of restricted diet, code of conduct, change lifestyle, and use of rasayana. I'm going to talk about rasayana. Rasayana is a food supplement. Yeah, which is very important. Okay. Oh, sorry. I'm going forward. Remove the cause and the disease. I have told you. So, how to do it? The balance your Agni, body clock, nature clock. There should be synergism with this. Rasayana and Biomedical prevents aging, gives longevity, strengthens body, and destroys the disease. So, you know, the peaceful and calmful, this was our traditional way, and now we are not following it, so we are getting into disease. Ahar Nidra Brahmacharya Yoga. What I mean to say that Dr. Sharma said that aging is not a disease, but Ayurveda says aging is a disease. They have told the Jara Vyadhi, they have used the word Jara Vyadhi. Means if a disease is there, a treatment is there. And for the treatment, they have recommended Rasayan. Rasayan is not only the supplement, there is Ahar Rasayan, there is Achar Rasayan, behavioral change. Everything is covered in that. So what I mean to say that what happens to these factors? If you all the three causes of diseases, there are 8 factors of diet, Langhan, psychology, gut microbiome, and circadian rhythm. If you are following these things, then only you are controlling your cell cycle. The imbalance of 6 dosas lead to the diseases. So what I mean to say that if you are following the lifestyle and you are not allowing the disease cause, then you will be healthy. This slide shows you the Agni. Agni means digestive power. There are 13 types of Agni in the body, but not much science has been done. But main is the Jatra Agni, where everybody knows that if your digestion is good and your health will be good, better. So Agni, long life, healthy life, and high Agni. Low Agni means disease. And this is related to the your GIT. And Prakriti, psychological, I have told you, Prajna Parad. So hyper, Rajas, Sattva, and Tam. So this is correct. There are 8 emotional states, the Krodh, Lobh, Moh, which is given in the Yam and Niyam. You should follow that. Then only you can be in the equilibrium of this. And psychosomatic disease. As I told you, 60% diseases are the psychosomatic. So you have to control your psyche. This is the computer. And this is the science which has been now proven. There is a relation between gut and your brain, gut-brain relation. And this is, brain is governed by hypothalamus, and these 5 sense organs are there. They are person to brain. So excess use of sense organs goes to the brain and then goes to your intestine, and both collectively cause the disease. So you have to take care of it. This is the cytone I made. Remove the cause, I told you. What are the cause of disease? Hurry, worry, and curry. The free radical theory, Dr. Sharma has told you, all the 3 things are causing the free radicals. Hurry means mitochondria is a powerhouse, you know. अगर कोयला जलाएंगे तो धुआं निकलेगा ही. कोयला तो जलाना ही नहीं तो खाना नहीं बनेगा. धुआं के लिए चिमनी बना देते हैं. इसी तरह से माइटोकॉन्ड्रिया में एटीपी तो बनाना है, नहीं तो आप कैसे जिएंगे. जो सुपर ऑक्साइड निकलेगा वो ज्यादा ना निकल जाए, उसके लिए सुपर ऑक्साइड डिस्म्यूटेज, कैटलेज, पेरोक्सीडेज, lot of antioxidant enzymes are there. So we have to manage a balance. The excess work, then your stamina also causes a disease. तो ये है, हरी हो गया. और वरी क्या है, कैटेकोलामीन जो निकलता है वो फ्री आयरन से रिएक्ट करके फेंटॉन रिएक्शन से हाइड्रोक्सी रेडिकल बनाता है. दिस इज अ वेरी रिएक्टिव फ्री रेडिकल. इसलिए कहते हैं अरे इनको तो चिंता खाए जा रही है. चिंता क्या खा रही है? चिंता ऐसे खा रही है कि ये फ्री रेडिकल बना दे रही है और आपको डिजीज कर दे रही है. और करी जो है, रोज हम तो जो फ्राइड फूड खा रहे हैं, डीप फ्राइड फूड, दैट इज द डायरेक्ट लिपिड पेरोक्साइड्स ऑयल वी आर ईटिंग एवरी डे. फास्ट फूड जो खाते हैं, क्योंकि घर में तो है एक ऑयल बना, एक बार पूरी छान दिया, और दुकान में वो कई बार दिन से रात तक पूड़ी छानते रहते हैं, तो ऑयल ऑक्सीडाइज हो जाता है, वही आप खाएंगे, इट विल कॉज डिजीज. So this is a cytone I have made. If the chimney is strong, antioxidant in your body is strong, then all vegetation is good. If you have an industry and the chimney is leaking, all smoke will come there and all vegetation will go there. So आपको जो सेल का जो एंटीऑक्सीडेंट है वो स्ट्रांग बना के रखना है. He have, अगर नहीं करेंगे तो क्या होगा? There is accumulation of arm. इसी पे आ गया था वो जो कि autophagy word. Arm means undigested protein, undigested food, or undigested metabolites in your cell. So ये जो है, there are two types, immature and mature. Like amyloid bodies, that is a mature arm. The immune complex, ये सब है amyloid. हम लोग इसको आम कह दिया, आयुर्वेद वालों ने कह दिया कि आम को हटाना है, आम का पाचन करना है. कैसे पाचन करेंगे? अग्नि बढ़ा के. There are 13 types of अग्नि. सबको बढ़ाना है. बढ़ाने के लिए लाइफ स्टाइल एंड फूड सप्लीमेंट दोनों. ये जो है लंघनम बता दिया आपको. लंघन इज द बेस्ट वे ऑफ बॉडी प्यूरिफिकेशन. इसीलिए हमारे यहाँ देखिए एकादशी का व्रत हर 15 दिन पे आ जाता है एकादशी. अगर महीने में 2 बार आपने व्रत कर लिया तो आपका बॉडी खुद ठीक रहेगा. नहीं कर पा रहे हैं तब आपको पंचकर्म कराना पड़ेगा. दोज हु आर नॉट एबल टू डू दिस थिंग, तो वमन विरेचन बस्ती नस्य. ये भी देखिए मिस नंबर हो गया है. इफ यू गो टू एनी 5-star होटल दे राइट पंचकर्मा, but they are doing what is power, only स्नेहन and स्वेदन. That is not पंचकर्म, that is पूर्व कर्म. तो ये जो 5 कर्म है ये पंचकर्म है, इसको करवाएंगे तब बॉडी प्यूरिफाई होगी. स्नेहन भी होगा लेकिन नॉट लाइक दैट if you want to do the arm. So फूड सप्लीमेंट, रसायन एवं वाजीकरण जो था ये बता दिया आपको. अब एक ही पॉइंट बताना चाहते हैं. यस. तो आचार रसायन, न्यूट्रिशन, and then vitality and longevity, काम रसायन, and nematics and disease oriented. There are 3 kind of rasayan. You have to follow that. This is one thing I wanted to tell. This is from the Ashtang Hridaya. जैसे हमारे यहाँ कहा कि जरा व्याधि कह दिया, तो जब आप पैदा हुए तभी से जरा व्याधि शुरू होगी, 100 साल तक रहेगी. तो चरक ने क्या किया कि every 10 years they have given a list of medicinal plants that you have to follow. Because you know, for example, the 41 to 50, the skin wrinkles will be there, 51-60, eye problem. They have given a word jatamansi. So what I mean to say that they have very scientifically defined it, these kind of things to follow. So this is what work I have done, different medicinal plant, and because the time is short, mainly I want to tell you that the Tamal is the Tej Patta, Pura tibrosa is the Bidari Kand, Nigella sativa is the Mangrel, and the Vitex nigundo leaf I have done. These are all the 30 PhD scholars have worked on this point. So this is the concept of heart disease. You know, the collagen cap is there. As long as collagen cap is there, the heart attack won't be there. When it becomes loose, you heart attack chance is there. So this is the BHUX which got the US patent, and it's a DBT funded project. We found that it is working on many targets. So it is also a plaque, this MMP is inhibitor. Because MMP is the cause which dissolves the catalase. This is the work on asthma on the Shirsadi Kasai of Ayurveda. And this is the mechanism of action YST and a mast cell stabilizer. You know, histamine and on modern medicine, antihistaminic drugs are given. We are giving the mast cell stabilizer. This is the Bidari Kand, and very quickly. This is the mechanism you say. I told you, hypoglycemic, antioxidant, anti-inflammatory, anti-apoptotic, and MMP activator. All the 5 things we have all scientifically validated through the molecular biology tools. And this is the mechanism we have come for the PTY2. You see, all these research papers, and this is the mechanism. What I mean to say that one plant is acting on many, many targets. See, Tamal, what I think Dr. Sharma said, I am also done this kind of work. If you are using insulin, you see, 0.01 unit, 11% decrease. And this 120 milligram per 100 gram, 21% increase. If I combine these two, there is a 59% decrease in blood sugar. So, I mean, if you are adding allopathy and modern medicine, there is an effect. This is Rubia cardifolia. This is anti-cancer, the best cancer model we have done. You can see the size of decrease. And lastly, this is the Tamra Bhasma work I have done. And then, this is the nanoparticle. The Manjista, which I told you, which I told you that Manjistha has a very strong antioxidant property. But when we make the gold nanoparticle of Manjistha, 38% fold in a therapeutic, in a macrophage cell culture, we found it inhibits the LPS inducing. What I mean to say that a lot of biotechnology is needed in this kind of, in validating the Ayurveda. With these words, I extend sincere thanks to all the chair and to the audience.
Thank you, sir, for that wonderful talk on Ayurveda. We can have few questions, please. Yes, sir.
Thank you very much, Professor. It's very nice talk and very comprehensive. So it made me understand the concept of Ayurveda much more. I woke up late today because I want to have enough sleep. Hope I get healthy, like you said. The thing that's quite popular in Thailand now, especially in young people working in the city, what we call intermittent fasting, or IF. It's related to starvation that you said in the lectures. So my wondering is what do you think about IF concept? Is it better to put it to be a lifestyle modification for the metabolic syndrome that you're talking about?
You mean the iron concept?
IF, in intermittent fasting.
Intermittent fasting, sir.
Okay, intermittent fasting. You know, the intermittent fasting in Ayurveda, they say that the time of clearing of food from stomach to intestine, that has a physiologically, it is already modern physiology, 2 to 3 hours. But they said they have linked to the hunger. If hunger is there, it means it is a symptom that your stomach is empty. Then you are eating. Then all your digestive system goes intact. So the intermittent fasting, the modern medicine, a lot of research paper has come. But in Ayurveda, they have told since the very beginning that hunger is the symptom. When you are hungry, you eat. That is a sign of intermittent fasting. Not like the 16-hour fasting, 12-hour fasting, because it's a personalized medication. For me, it may be 8 hours. For you, it may be 14 hours. When you are hungry, it means it is intermittent. No question.
Sir.
Sir, one more question.
Sir, good morning, sir. Good morning, sir. सर जैसे सभी ड्रग्स का अपना अपना एक साइड इफेक्ट्स होता है, तो क्या ऐसा युग में भी है कि अगर हम जो एक्सरसाइज पर्टिकुलर बनाया गया है, अगर उसको एक्सरसाइज करते वक्त उसको गलत तरीके से परफॉर्म कर लें, तो उसका भी कोई साइड इफेक्ट्स है हमारे बॉडी पर?
एक्सरसाइज का,
योग का सर।
योग का, देखिए योग जो है ना, उसको लिखा है स्वांत सुखा है। जितना आपका जो आपको सुख दे वो योग है। जिम इज ए स्ट्रेनुअस एक्सरसाइज। तो जो पैरासिम्पैथिक और सिम्पैथिक नर्वस सिस्टम है वही यहाँ पे काम करता है। जब आप अपने सुख के साथ योग करेंगे तब आपको सिंपैथेटिक नर्वस सिस्टम कम होगा, पैरासिम्पैथिक ज्यादा होगा। जब जिम में जाते हैं तो वो जिम ट्रेनर हमको ज्यादा ट्रेनिंग करा देता है, तो सिंपैथिक नर्वस सिस्टम में कॉर्टिसोल जो है वो बढ़ जाता है, तो वो बहुत नुकसान कर देता है कभी कभी। इसीलिए कहा गया है कि यू हैव टू वर्क इन इक्विलिब्रियम विथ योर बॉडी। अच्छा, ये भी कहा गया है आयुर्वेद में कि कितना एक्सरसाइज करें, जब माथे में पसीना आ जाए और काफ में पसीना आ जाए तो एक्सरसाइज बंद कर देना चाहिए। अब ये देखिए, ये सीजनल चेंज है, जाड़े में देर में आएगा, बरसात में बहुत जल्दी आएगा, तो बरसात में कसरत कम करना चाहिए और जाड़े में कसरत ज्यादा करना चाहिए। सब सिम्टम लिख दिया आयुर्वेद वालों ने, लेकिन हम लोग उसको साइंटिफिकली अब लिंक नहीं कर पाए। दिस इज द पॉइंट।
थैंक यू।
यस,
I think I just want to add, like, no, you mentioned that the aging is a disease. No, so no disrespect, but I disagree with that. Aging is not a disease. Aging is a risk factor for many of the diseases we study.
Okay, I am not saying aging is disease. It is a Charak who has said, Jara Vyadhi. I am not saying. Maharishi Charak has said, and you know what is the difference in my feeling and your feeling? If you have a bulb of 0 degree, you see only this much. If you have a bulb of 100 power, you see a long distance.
What is required? Jara Vyadhi, Charak ने कहा
उसका एक्सप्लेनेशन आप ढूंढिए। जरा व्याधि का मतलब यह है, हमारी समस्या, आप लोग तो विद्वान हैं, कि मतलब बुढ़ापे में बहुत सारी चीजें हम नहीं कर पाते हैं। हमारी मसल स्ट्रेंथ नहीं होती है, हमारी लंग कैपेसिटी नहीं होती है, हमारी आंख काम नहीं करती है, ये व्याधि है। तो जरा व्याधि जो उस समय रेफर किया गया, हम चरक संहिता का मैं विद्वान नहीं हूँ, बट आई थिंक उसको उस तरह लेना चाहिए।
अच्छा,
ये नहीं कि एजिंग इज ए डिजीज। व्याधि का मतलब कि आपको बाधा पड़ती है।
Okay.
Right.
Okay. I have
Just a second. Aging definitely is not a disease. However, aging is a risk factor for several diseases.
No, what Dr. is saying, you are saying the same thing. But what I am saying, because Ayurveda has its two objectives, swasthasya swas rakshanam, atrus vikasa prasannam cha. Means if aging is a risk factor, then it should be managed from the very beginning. Not like after 65, when you become old age. So you should start preventing aging. The premature aging is very important nowadays. That is why to prevent the early aging, you should start from the very beginning. This is why they have told Jara is a Vyadhi, means it may be a risk factor for the many diseases, but you should start preventing it. Because the main objective of Ayurveda is swasthasya swas rakshanam. When you know it is risk factor, why not to start preventing it by taking your lifestyle change?
I think that's what, like now in modern days, we say healthy aging. So you want to do everything good to you age healthy.
Yeah.
You should not develop many diseases.
That's right. That's right.
We cannot, no matter what we do, how much science we can develop, we are not going to be able to prevent aging.
No, but there is a word has come, premature aging, right? The premature aging can be prevented, can be delayed. Aging cannot be done, but premature aging. But mostly people are now premature. He said very rightly, you want to increase the age in your life or you want to increase the health in your life. So health in your life is the premature aging prevention. That is why he said the Jara Vyadhi. You know, the Ayurvedic people were very intelligent. They've given one shloka. You keep on writing a book on that. So we are not going to on that controversy, because they are criticizing Ayurveda will not help in validating Ayurveda. What I used to always say, you have a 100 power bulb and you have a 10 watt bulb. So today we have a 10 watt bulb. What the Langhanam MAHAYOGI was given, autophagy has come in 2016, Nobel Prize came. So tomorrow or today, it may come, we don't know. As a scientist, I cannot claim it. As you are saying, I'm also with you.
So I guess this is an endless debate. So due to time limit, we have to stop. We will have the, okay, one last question I ask.
Thank you so much, sir, for the nice and the comprehensive lecture. So my question is, as you are interplaying between the Ayurveda and the allopathy, so how you choose the drug targets when you are checking the crosstalk, means when you are choosing two different sciences, because like in case of the computational biology, we know that computational biology chooses the drug target on the basis of the allopathic things. Like in any diseases, you are using both of the things, like Ayurvedic science as well as the medical science, allopathic sciences. So how you are choosing the drug targets? This is actually, I am not aware of this thing.
Very nice question and very pertinent also, because my title was the integrative approach. For example, the inflammatory, please sit down. Inflammatory disorders, you know, the main metabolic syndrome is a systemic inflammation. Your body inflammation starts going on, which can be monitored by HSCRP, high sensitive C-reactive protein in your blood. So if it is going high, in the aging also is a inflammatory process, and obesity also is an inflammatory process. In both the conditions, modern medicine has a set guideline for the treatment. But what Ayurveda says decrease the Vata. Vata means some comparative study has been done. Vata has been correlated with the inflammatory markers. So IL-6, NF-kappa B activation, these things are happening in your obesity. This is happening in your aging, old age. So if this can be prevented by your modern tools, so we can add antioxidant of your spices, your herbal supplement. This is the way. So what we are targeting the Vata, Pitta and Kapha. And Vata has been linked with several biochemical markers, and more research is required. And up to now, we have come only one conclusion, that the inflammatory markers are increasing with Vata. So we are selecting all the medicines which are given in the Vata decreasing, and we are increasing those medicines which the Kapha increasing. So increase the Kapha and decrease the Vata. This is like a titration. And Ayurveda has given a set of 500 medicinal plants have been listed in Charak in 50 decoctions. They are called Mahakasaya. Like Medhya Rasayan having 10 medicinal plants for brain. They are not saying that there are the 50 or 100 neurological diseases, but they are saying in all the diseases, give Medhya Rasayan. How it is working? This is the bioinformatics will work. You understand? Yes. How we are combining? We are combining Vata reducing drug and Vata increasing drug. And Vata has been linked to the systemic inflammation. This is
okay, sir. We'll have to wind it up. Thank you, sir. Thank you.
Thank you, sir, for your enlightening lecture on Ayurveda. Your insightful discussion on Ahar, Nidra, and Rasayana had provided us with a deeper understanding of the fundamental pillars of health and longevity. Now, I request Professor Edithil Vijayan, sir, to kindly felicitate Professor Sarita G. Bhatt, madam, and Mr. Sujit Nair, sir, chair and co-chair for the sessions. May I now invite, sir, Professor Dr. Edithil Vijayan, sir, for the felicitation. Thank you, sir. May I now request Professor Dr. Sarita ma'am and Mr. Sujit Nair, sir, to kindly felicitate Dr. Rupali Gupta, madam. Thank you, ma'am. May I now request the chair and co-chair to kindly felicitate Professor Dr. Yamini Bhushan Tripathi, sir. Thank you, sir. Now it's time for tea break. Ladies and gentlemen, we'll now take a short tea break.